Hi guys, I'm Mrs. Elias again. If you didn't watch my video last week, I'm doing a tutorial over meiosis just a little bit and genetics. So we're going to talk about monohybrid crosses, dihybrid crosses, mostly Mendelian genetics, but we'll also cover a couple non-Mendelian things like incomplete dominance and codominance and sex-linked traits at the end of the video. So we're going to start by going over a couple things about meiosis, and really what you need to know for the STAR test about meiosis is the differences between it and mitosis, and then also crossing over. I think that's really it. You need to know why meiosis is different, what it makes, how many cells it makes, and then why crossing over is so important. So here's a picture of the steps of meiosis. And so hopefully you watched the mitosis video, I think Mr. Cotton did a couple weeks ago, or you just remember. But we have prophase, metaphase, anaphase, telophase, and cytokinesis. So the phases are in the exact same order, and almost the exact same thing happens in them in meiosis. But what's happening in meiosis is that we are making 4 cells, and they have half the number of chromosomes as the original diploid cell. So there's a couple of vocab words we need to talk about. Diploid and haploid, these are the words that we use to describe the amount of chromosomes in a cell. And I'm going to use humans as the example. So our human diploid number of chromosomes is 46. So you get 23 of your chromosomes from your mom and 23 of your chromosomes from your dad. And together, those make your 46 chromosomes, which is your diploid number, or a human's diploid number, because most humans have 46 chromosomes, with the exception of some genetic disorders. The haploid number is the half number of chromosomes. It's 23. So meiosis is the cell division that makes gametes, or sperm cells, or egg cells. And sperm cells and egg cells have to have half the number of chromosomes, or the haploid number of chromosomes, so they can pass on half of their genes to their offspring, essentially. And then offspring get all 46 of their chromosomes by getting a sperm cell and an egg cell, which fertilize, it's the first part of development, from mom and dad. So you get 23 chromosomes from mom and egg cells, and 23 chromosomes from dad and sperm cells. So meiosis is only making sperm cells or egg cells. Mitosis is for creating somatic cells, or all of your other body cells that are not sperm or egg cells. And so we need these cells to have half the number of chromosomes. So in order to achieve that, we have to have 2 rounds of cell division. So we have meiosis 1, which takes our haploid cell, we replicate the DNA. So really we go from 46 chromosomes to having 46 chromosomes that each have a copy of themselves, which we call sister chromatids. They're attached to each other, which is why these look like little X's in the picture. Then after meiosis 1, we've divided those cells into 2 cells, which really have 23 chromosomes, 1 of each homologous pair, 1 of each kind of chromosome that you have. So because you get 23 chromosomes from mom and 23 chromosomes from dad, we have 2 copies of every chromosome, 1 through 22, and either an X or a Y chromosome. So each of these has those chromosomes in it. We've divided our homologous pairs in half, which is what happened here. If you see in this picture in my metaphase 1, we have the red version of this chromosome. So maybe moms are red and dads are blue in this picture, but these are 2 of the same chromosome 1, for example, but you have 2 copies of it. And they get divided by being pulled apart in anaphase and then separated in telophase into 2 separate cells. Then those cells go through another round of cell division. So again, prophase 2, metaphase 2, anaphase 2, telophase 2, the second time. And this is just dividing the copies of the chromosomes. So during DNA replication, a copy of every chromosome is made, and they are attached to each other until they're divided in this final step in meiosis 2. So meiosis 1 divides the homologous pairs apart. Meiosis 2 divides the copies or sister chromatids apart, so that each of these 4 new cells will only have 1 copy of 23 chromosomes. The other important thing to know about meiosis is during prophase 1 here, what's happening is all these homologous pairs of chromosomes. So the 2 copies of those chromosomes that we have, one from mom, one from dad, pair up and they cross over. It's easier to see here. You see there's a little bit of red on the end of this chromosome and a little bit of blue on the end of this one. You can kind of see the different colors on these ones. That happened because during crossing over, sections or little segments of the chromosome, which carry related or the same type of gene, switched over. Here, let me draw this on the board. So what's happening in prophase 1 of meiosis are homologous pairs are lined up next to each other. So let me define these real quick. So in DNA replication before any kind of cell division, whether it's mitosis or meiosis, your homologous chromosomes get a copy of them made. So each of these is a copy of the same chromosome. So this is a chromosome, this is another chromosome. These 2 chromosomes are homologous chromosomes. And what makes them homologous is that they carry the same type of information about the same traits potentially, or reality. So like if this is the section of this chromosome that has the eye color gene, and from one parent you get the blue eye color gene, and this is the same section on this homologous chromosome that carries the eye color gene, it could also be the blue eye color gene, or it could be the brown eye color gene, it doesn't matter, but you're getting one blue eye gene from one parent and one brown eye gene from another, or you're getting 2 eye color genes in general, whether or not they're, what kind of version of that gene doesn't matter. We'll talk about that in a few minutes. But homologous chromosomes carry the same information. They're always about the same size and shape. But these copies are called sister chromatids. So we call copies of the same exact chromosome sister chromatids. All right, and so your sister chromatids are held together by a centromere. We usually just draw them as X's. That's what's holding those 2 copies together. These are 2 copies of the same chromosome. So in crossing over, once this happens in prophase 1, after these guys get pulled apart in anaphase 1, they're going to have a little bit of the other chromosome's DNA on them, basically. They trade segments of DNA, and it probably would happen down here too, I guess, because they crossed over at the bottom also. But this is just a visual representation of what happens. So this chromosome now is not the same as it was genetically when it was over here and hadn't crossed over yet. And each of these copies is not going to be exactly the same either. What happens is now if we go to the last step where we separate the sister chromatids, we're going to have one that looks like this and one that looks like that maybe. And then we'll have the red ones. One looks like that, one looks like that. So because they traded DNA during crossing over, none of these, what started out as identical copies of 2 different chromosomes, these 2 are not genetically identical anymore. These 2 are not genetically identical anymore. So if you have none of your chromosomes that are genetically identical, you're increasing genetic variation. So the main thing to remember about crossing over is that it increases genetic variation. And when we get to evolution review, this will be very important. Genetic variation is really important for the survival of a species and for sexually reproducing organisms like humans and animals and plants. It's very important. So crossing over happens during prophase 1 of meiosis. This is when it happens. And then it increases genetic variation in general because now none of those chromosomes that are going to go into 4 different cells are genetically the same. All right, so crossing over is important to remember about meiosis, and then what it does. It makes gametes, sperm or egg cells, and it makes 4 of them, and they're haploid, meaning they have half the number of chromosomes. So I did the humans as an example, but if you use another animal, dogs have 78 chromosomes in their homologous, I'm sorry, diploid cells, like in their body cells. Their haploid cells would have half of that, which is 39. So you always have the diploid number is like 2 sets of chromosomes. Haploid number is just 1 set or half of the total number. So you should be able to go back and forth between diploid and haploid if given one or the other. So you need to know diploid means double, di means 2, haploid sounds like half. I don't think it actually means half, but that's the way I remember it. Okay, let me find a meiosis practice question real quick. I think there's one in here somewhere. Here we go. So during meiosis, homologous chromosomes exchange genetic material. So it's not using the term, but we know that this exchange of genetic material is called crossing over. That's when your homologous chromosomes, like it says here, swap a little bit of DNA. This exchange of genetic material increases genetic variation. Look at that. That's exactly what I told you. You needed to know about meiosis. They're almost always going to ask a question about crossing over and genetic variation. If it's not in the answer, it's usually the answer, guys. Meiosis doesn't reduce the diploid number to the haploid number. Well, meiosis does, but crossing over, that exchange of material, crossing over specifically, is not the whole process of meiosis, so it doesn't do this. This is describing all of meiosis. There is no process of increasing the haploid number to the diploid number because even mitosis just goes from diploid to diploid. And reduces the probability of mutations. Actually, crossing over probably increases the probability of mutations because we're literally changing some DNA. So the answer was increases genetic variations. The most important thing to remember about meiosis. All right, sexual reproduction in animals depends on the production of gametes. Which of these processes produces gametes in animals? So like I said, you just need to know what does meiosis produce? It produces gametes. And remember, gametes are sex cells or sperm or egg cells. So the answer is meiosis. Mitosis would be somatic or any other cell in your body besides sperm or eggs. I think those are the only 2 meiosis practice questions. So we're going to move on to genetics real quick. Let's do this slide first. We need to review some vocab. I'm going to raise my board. So when we're talking about genetics, you have to know the vocab terms homozygous and heterozygous. You won't be able to do these questions if you don't know these 2 vocab words. So I'm sorry that these are spelled wrong. This is not my original PowerPoint. I can't edit that. This should say homozygous, sorry. Homozygous means you have 2 copies of the same allele. Now let's talk about what an allele is. Remember when I was describing the genes that are carried on your homologous chromosomes? You get 1 allele from each parent. So like you get an eye color allele from mom and an eye color allele from dad. And maybe you get the blue-eyed allele, which happens to be recessive from one parent, and you get the brown-eyed allele, which happens to be dominant from the other parent. Those are your 2 alleles. And if you had 2 different ones, like I just described, those would be heterozygous, 1 dominant, 1 recessive, 2 different alleles. If you have homozygous alleles, that would mean that you got from your parents 2 blue-eyed genes or alleles, or you got 2 brown-eyed genes or alleles. If you got 2 copies of the recessive gene, like blue eyes, then that would mean you have a homozygous recessive genotype. If you got 2 copies of the brown-eyed allele, for example, that would mean you have a homozygous dominant genotype. And that's because brown eyes is dominant over blue. And in test questions, it'll either tell you what's dominant or what's recessive, or give you information to figure that out, one way or the other. So homo means same, so 2 of the same recessive alleles or 2 of the same dominant alleles, and then hetero means different, so 2 different alleles. All right, let's look at this practice question. This is just showing you how to fill out a Punnett square. So we use 4 boxes for monohybrid crosses, meaning we're following only one trait. So if you're reading a practice question and it's saying a man who's homozygous, or I'm sorry, let's do this practice, 2 parents who are heterozygous for maybe this eye color trait have offspring. What are the chances their kid would have such and such genotype or phenotype, or have blue eyes or have brown eyes, whatever. So let's say these are 2 heterozygous parents. That's why we wrote big R, little r, dominant, recessive. Remember, heterozygous means 2 different alleles. So these are 2 different alleles here. And then this parent also is heterozygous. So they have 2 different alleles, big R, little r. When you fill in your Punnett square, you just cross what would go in that box. On the top, we have a big R. On the side, we have a big R. Over here, big R, little r. Over here, big R, little r. Little r, little r. I'm going to do this on the board. Okay. So when you're looking at this, like if the question asked you, what are the chances that their kid would have brown eyes? Because dominant alleles are dominant, that just means they cover up. If you have a copy of one dominant allele, it's always going to cover up the recessive allele. So even if you have a copy of the blue-eye gene, because brown eyes is dominant, it's going to cover up that trait in your phenotype. So this kid's going to have brown eyes, this kid's going to have brown eyes, and this would have brown eyes. So there's a 75% chance of having brown eyes in this case. And then that leaves 25% chance that their kid would have blue eyes. So the way I describe this, brown eyes and blue eyes, these are what we call phenotypes. Remember, phenotype. Phenotype means physical appearance. So like I could physically see they either have brown eyes or blue eyes. And then genotype means their genes or their combination of alleles. So a homozygous dominant genotype gives me brown eyes, but a heterozygous genotype also gives you brown eyes. Only homozygous recessive genotypes give you the recessive phenotype, all right? There's a lot of vocab in this unit that you have to remember. Back here. All right, so this is just a regular old monohybrid cross using Mendelian genetics. And when I say Mendelian genetics, I mean dominant always covers recessive, okay? Only in specifically Mendelian genetics. And you can always assume it's that unless it specifically says incomplete dominance or codominance, and we'll talk about those in a few minutes. Okay, the last thing we need to look at, not the last thing, the next thing is dihybrid crosses. I'm just going to use this example of seed shape and seed color in my practice. So we're using R's and Y's. I'm just going to use on the board. Okay, so is it color? Seed shape, seed color, seed shape, seed color. Okay. Oops, I think wrinkles actually are recessive. Round seeds are dominant. Wrinkled seeds are recessive, little r. And then we're going to use big Y. Doesn't look like a big Y. Big Y is for yellow seeds. And little y is for green seeds. Okay. Let's pretend that we're going to cross two parents who are heterozygous for both traits. We're doing a dihybrid cross. Dihybrid means two traits. So we're following, this is like trait 1 is type of seed or seed shape. Trait 2 is seed color. We're following two traits at the same time. What are the chances of inheriting, both of these in different combinations? So when we write a genotype for a dihybrid cross, we have to have 4 letters. You're gonna have 2 for this gene and 2 for this gene, because remember, you have 2 copies of every gene because you have homologous chromosomes, one from mom, one from dad. So if we're crossing 2 heterozygous parents, that means they're heterozygous for both traits. What we have to do, I'm going to show you guys how to do the big 16 box Punnett square first, and then I will show you the faster, maybe easier for some of you who like fractions version of the fraction method. So first thing you have to do is FOIL your genotype. Because both parents have the exact same genotype, I'm only going to do it once. But let's say you had 2 different genotypes. You'd have to FOIL each individual parent's genotype. And so when I say FOIL, we're looking for all the possible combinations of their alleles for both traits. So let's say we would have, they could possibly give their kid big R and big Y. They could give their offspring big R and little y. They could give their offspring little r and big Y. And they could give their offspring little r and little y. Both parents are the same, so if I FOILed this, it would be the exact same 4 possibilities. Basically, like if this is the mom plant, she could give her kid this in one of her egg cells, or this in one of her egg cells, or this, for example. So once you FOIL the genotypes to get all the possible combinations of those alleles, you're going to make your 16 box Punnett square. You take those combinations that you just made, and because we have the same parent, you're going to put one on the top, one on the side. If this parent had a different genotype, no, I want to do it. Okay. If this parent had a different genotype, you would put its on top and then this one on the side. You separate the parents, but this one, they happen to both be the same. Okay. So I put all the combinations on one side for each parent. Then just like a monohybrid cross, you follow what's on top and on the side of each box and you fill it in. So we have big R, big R, big Y, big Y. It's important to always put the letters back together. Put the R's back together. Don't do big R, big Y, big R, big Y. It's going to be harder to read, to find out what the genotypes are. So put your R's back together, big R, big R. Big Y, little y. Big R, little r. Big Y, big Y. Big R, little r. Big Y, little y. I made a mistake there, little r, little r. Big Y, little y. Okay, after you do all of that, we have all the different possible genotype combinations. Remember, your genotype is your combination of alleles. And for a dihybrid cross, every individual has 2 copies of each trait, so 2 traits, 4 alleles total. On a STAR test, they're not going to ask you for all the possible genotype combinations or all the possible phenotype combinations. They're going to ask you for one more than likely. So let's say the question asked us, what were the chances of getting big R, big Y, little y as the genotype from this cross between these parents? So what are the chances their kid has big R? Oh, you can't see that, it's off the screen. What are the chances the kid has big R, big Y, little y as their genotype? So you look on here for how many out of 16 are this genotype, and you should find 1, 2, 3, 4. When you do a dihybrid cross with 2 heterozygous parents for both traits, the chances of getting that same genotype is always 1/4, or 4 out of 16, or 25%. No, 25, they go next to each other. 25%, okay? It could also ask you for a phenotype. So what are the chances of getting a kid or a baby offspring that has round seeds? Since we're talking about plants, it's weird to say baby. Offspring with round seeds, and the second trait, let's say green seeds. So what are the chances of getting round and green offspring? So we're looking for what are the chances of getting round seeds and green seeds. Now green is recessive, so none of them that have a capital Y are going to be green. So we can eliminate any of them that have a capital Y in them, because it's not possible to get a green seed with a capital Y in their genotype. So that gives us these 4 that are all recessive, which give us green. Oop, not this one. So 3, 4, there's the 4th one. But then round seeds. Now this is any of them that have at least 1 capital R because it's dominant. So you only need 1 capital R to have a round seeded plant. So the only one we're also going to eliminate would be this one. So that means there's 1, 2, 3. 3 out of 16 chance of getting this specific phenotype. So remember, you can look for phenotypes or genotypes when you do a dihybrid cross like this. Now I want to show you the fraction method or way of doing this, the same question. We're going to rewrite this, erase all this work. If you're good at multiplying fractions, this might be the best way for you to do this. Basically, what you have to do is just 2 monohybrid crosses. Because of Mendel's laws of inheritance, you inherit all of your traits separately, or we assume that at least. So you inherit, these plants would inherit the shape of their seed trait separate from their color of their seed trait. So we can look at individual monohybrid crosses for this trait with these parents. So you take the 2 parent genotypes for one trait, you make a monohybrid cross. Just like we practiced in the other, we did monohybrid crosses. Then you do 1 monohybrid cross for the second trait. All right, so we could take the probability of getting this trait and the probability of getting this trait and multiply them together, basically. So if I wanted to still know what were the chances of crossing these parents with these 2 traits and getting this genotype, I can do that. So you look at your Punnett square. What are the chances of getting big R, little r? You've got 2 here. So that's 2 out of 4. Then what are the chances of getting big Y, little y? Again, I've got 2 here, 2 out of 4. So I'm going to multiply these fractions together because it was 2 out of 4 times 2 out of 4. When you multiply fractions, you multiply across. 2 times 2 is 4. 4 times 4, 16. So again, same answer is 4 out of 16 or 1/4 or 25%. If you do this, you need to be able to go to percentages. The STAR test loves to put them in percents. So be able to do that. You do have access to a calculator on the biology STAR test if you need it. And so you would divide 4 by 16 to get 25%. Okay, we can do the same thing for round and green seeds. So all you need to know is just how the genotypes turn into phenotypes. So for round seeds, if I'm looking at this Punnett square, that's a different color. I want to know what are the chances of getting round seeded plants out of this. So all I need is 1 capital R. So that means I have 3 of these that give me round seeds. So 3 out of 4 is our fraction. 1, 2, 3 out of 4. What are the chances of getting green seeds from this one? Remember, green is recessive. So that means only 1 gives us green seeds. So we multiply the probability of getting round times the probability of getting green, and that gives us our chances of getting both round and green. 3 times 1 is 3. 4 times 4 is 16. Don't look at this and think, oh, it's just 1. It's 100%. Don't add the fractions. Make sure you're multiplying the fractions. So 3 times 1 is 3. 4 times 4 is 16. You add the numerator and add the denominator, which is the same answer we got last time, if you remember. So if you do a big dihybrid cross or fraction method, you're going to get the same answer, which is important to know. Okay. Let's look at some practice questions now. So in Cocker Spaniels, the allele for black coat color is dominant over the allele for brown coat color. If a Cocker Spaniel is crossed with a heterozygous black Cocker Spaniel, which of the following genotypic ratios can be expected? I'm gonna show you on the board what I would do if I was taking this STAR test and needed to answer this question. You guys have 4 hours to take this test, and it's more than enough time. So never feel like you have to rush because you have plenty of time. So I recommend always filling out a Punnett square, even if you think you're sure. So what we're gonna do, we're crossing a brown Cocker Spaniel and a heterozygous black Cocker Spaniel. So hetero means different. So with the heterozygous black Cocker Spaniel, we know their genotype has to be big B, little b. 2 different alleles. And then brown is recessive to black from the question, that's what it said. So we're going to cross, it said a brown Cocker Spaniel, and now brown's recessive. So that means for that dog to actually be brown in its phenotype, it has to be homozygous recessive in its genotype. So then we fill in our Punnett square. Okay. So I'm going to go back to the answer choices. Any of them that have big B as an option are wrong, because when we did our Punnett square, if you can see up in the corner of the screen, none of these gave us big B or a homozygous dominant genotype. So that automatically gives us F, 2 big B, little b, and 2 little b, which is what we got. We have 1, 2 big B, little b, and 1, 2 little b's. All right, this is a ratio. They're just showing you how many out of 4 was each genotype. All right, let's look at another one. This is a dihybrid cross. How did I know that? Because we're looking at facial dimples and free earlobes. These are two different traits. Both facial dimples, or having dimples, and having free or not attached earlobes are dominant. What are the expected phenotypes of the offspring of a female with dimples and free earlobes? They give you her genotype, which is very nice of them. They don't always do that. And a male with no dimples and attached earlobes. He's recessive for both traits. So they want to know, what are the chances that these two parents, or they want to know, what are the expected phenotypes? Okay, so what's all the possibilities their kid could be with these two parents? So I'm going to do the fraction method because it's so much faster. But if you're not comfortable with it, when you do your own, STAR test, make sure you do which one you're comfortable with. Because remember, time's not a problem for you guys on the STAR test. So we're crossing a mom who's big D, big F, little f, because that's what they said she was. And then a dad who's little d, little f. Okay. So looking at this, I'm going to do the fraction method. If you were going to do the big 16-box Punnett square, you have to FOIL this, and then you have to separately FOIL this to get what goes on both sides. But I'm going to do the fraction method. So I'm going to do one monohybrid cross for the D trait, which, because they're both homozygous for both their traits, the only option is to get big D, little d from these two parents. He can only give big D, she can only give little d. Then I'm going to do a cross for the earlobe trait, I think it is. So big F, little f from dad, and then little f, I'm sorry, mom, dad, whatever. So you fill in this Punnett square. Okay. So looking at this, there's 100% chance their kid's going to have dimples, because dimples are dominant. So 100% chance their kid's always going to have dimples. There's a 50% chance they'll have free earlobes, and a 50% chance they'll have attached earlobes, which is the recessive version. This is attached, this is free. And actually, I'm going to put these into fractions. So this would be like 4 out of 4, and this would be 2 out of 4, 2 out of 4. Because this question asked us for phenotypic possibilities. So looking at this, answer choice F says 50% chance their kid would have dimples and free earlobes, but actually we can eliminate 3 of these already. So let's just read through all of them. 50% with dimples, free earlobes, 50% with dimples, attached earlobes. 50% with dimples, okay, that's still free. 50% with no dimples. Is it possible from our monohybrid cross for any of their kids to not have dimples? No. All of them have dimples no matter what their ears look like. So any answer choice that says no dimples, we can eliminate. That's 1, 2, 3. We can eliminate G, H, and F just because we know all of them are going to have dimples because mom's homozygous dominant. So that means F is the correct answer. 50% of them are going to have dimples and free earlobes, which we had the 50% here. So dimples and free earlobes. And then 50% are also going to have dimples and attached earlobes. Those are the 2 options. So the answer is F. I think we have one more practice question. It is also dihybrid. In the 1860s, Gregor Mendel performed numerous dihybrid crosses between pea plants. Dihybrid crosses involve a study of the inheritance patterns related to 2 different traits. They actually defined a dihybrid cross for you. In guinea pigs, the allele for black fur is dominant over the allele for brown fur. And the allele for short fur is dominant over the allele for long fur. What percentage of the offspring from this cross between these two parents, a heterozygous parent for both traits and a homozygous recessive parent for both traits, would be expected to be heterozygous for both traits. So basically, we want to know when we cross these two parents, what % of their kids are going to be this, okay? Again, I'm going to do it in the fraction method. So we're crossing one parent who's heterozygous. Didn't use F's, I forgot. Yeah, okay, sorry about that. And then a parent who's homozygous recessive for both. That's a capital. Nope, no, it's not. Okay. So when you do the fraction method, you just do one monohybrid cross for the B trait or the hair color trait, I think it is. Big B, little b for this parent, little b for this parent. Okay, then you do one fraction or one monohybrid cross, sorry, for this parent. Big F, little f, little f, little f, or that trait. Okay. When you fill that in, you get big, big, little, little. Okay. So the question asked us, what are the chances or what's the probability of getting this genotype, heterozygous for both traits? So looking at this Punnett square, what are the chances of getting big B, little b? 2 out of 4. Times whatever the chances are for getting big F, little f. So from our monohybrid cross, we have 2 out of 4. When we multiply these probabilities together, because they're fractions, you multiply the numerators together. 2 times 2 is 4. And then you multiply the denominators together. 4 times 4 is 16. You can reduce that to 1/4. And then you also will have a calculator on the STAR test. You could just divide 4 by 16 and get 25%. So let's go back to our answer choices and see if that was an answer choice. It should be 25%. All right, yep, that was it. All right, I hope you found this tutorial helpful. Make sure that you use take the practice quiz that goes along with this to earn your bonus points. Thanks for watching. Bye, guys.